For that introduction. Good evening, everyone, and thank you for sticking around. It's quite a long evening for you all, and thanks for inviting me to speak. I'm going to talk about two projects that I'm currently working on, but first I thought it would help to give you some context around my practice as an artist. So I'll spend the first 5 or 10 minutes on that and the second half of the talk on my new projects. This slide is not one of my works, but it does kind of sum up what interests me. I originally studied psychology, and this kind of image here, this process, I don't really see it as circular, but more of an ongoing spiral or a vortex that's changing all the time. And I love thinking about the idea that the current manifestation of this evolutionary cycle with humans at the summit may well give way to something else in future millennia. Or, perhaps there are even other parallel vortexes. Really, the natural world has always been a key focus to me in my research. And so our interactions as humans within it and our impacts on it are important in my work. Now, originally, this kind of manifested in my practice as an exploration of consumerism and waste, inspired by pop art, the art of a Vera group, and writers like Jean Baudrillard. I began by making these large sculptural rabbits that are made entirely of waste materials. I've been researching Fibonacci's number sequence, which is based on the pattern of rabbits breeding. And I liken this analogy of breeding rabbits to our accumulation of waste. So I had this notion of these rabbits born out of waste, feasting on our accelerating consumerism and multiplying everywhere. I then started to look more directly at our relationship with nature and focus instead on human technological progress and scientific research. I was experimenting with the cyanotype print process because I loved its rich scientific blue, and their use as blueprints in the 19th century referenced the DNA and the genetics that I was researching at the time. And during this time, I came across cyanobacteria, and that led me in a new direction, using live materials in artwork to focus on our symbiotic relationship with nature. So I became interested in how microbes are being studied in the lab and the many ways in which genetic engineering and synthetic biology are being used to adapt life. And I started to explore humanity's use of technology and biology to enhance us as a species. We've evolved to this point where we can deconstruct our very being at the molecular level. And I began to reflect on what might exist beyond human. So when I created this work and its counterpart, which you'll see on the next slide, I work with artists, scientists, and philosophers. In fact, one of the scientists is here tonight, I think, Chelsea, somewhere, to create a publication and an audio on these themes of being beyond human, which went alongside this celestial-themed exhibition of live cyanobacteria and microalgae shown at the Hatton Gallery in Newcastle. So this really brings me to the beginning of my current research, where I take on... Oh, lost it. Can you all hear me okay? Yeah. Where I take on the role of the curious animal and explore an area of research where I think the human drive for progress is particularly manifest, and that is synthetic biology, the 2.0 of genetic engineering. Martin Heidegger in 1955 quoted American chemist Wendell M. Stanley, who predicted that the hour is near when life will be placed in the hands of the chemist who will be able to synthesize, split, and change living substance at will. And well, today this hour has passed, and life is in the hands of not only the chemist, but the computer scientist, the engineer, the designer, and the artist. And in the same address, Heidegger spoke of a potential for imbalance in our technological era between what he described as calculative thinking, which I equate to a reductionist scientific approach, and meditative, more reflective thinking. And in my research, I'm attempting to focus on the latter in the context of biotechnology. So I began to look at the life of the microbial other. Knowing as we do now that bacteria can communicate through quorum sensing, I started to think about them as a kind of swarm intelligence operating across deep time. So I'm making work that attempts to understand and possibly even respect them more, which led me back into the lab. And I like this quote from Nam June Paik because I don't think he hated technology. I think he was fascinated and repelled by it at the same time. And that's how I feel about synthetic biology. It's human nature. We're curious animals. We choose to work with life as this reducible material because we can. Yet as soon as we do, we're using the living as standing reserve, as tools for our purposes. And I believe that there's something about the simple cell which has the ability to adapt to its environment and pass that knowledge to future generations, a communication through evolution, if you will, that gives the microbial a kind of presence beyond time, a sort of stored knowledge of the future. So for me, life, even as simple as the cell, has an agency or an essence that through technology we can come to understand more, but perhaps never fully. So I'm going to play you a short video clip now from an installation that I made as part of Lumiere Durham last year. And you're just going to see the film, so you have to imagine that you're outdoors in the freezing cold of a Durham November and looking at this folly in a Greek temple. Hopefully the sound will work with this. Sorry, the sound was very quiet. I don't know if you could hear it very well at all, but did you just about pick it up? Okay. So what you've just seen and heard are a microalgae called Dunaliella salina. Scientists are more likely to look at this view, or even a higher order of magnitude inside their bodies. And by and large, the scientists that I've worked with don't listen to them at all. These microalgae are scientific material that we exploit for their potential use as fuel, food, and medicine. But when I saw them through the microscope for the first time, I didn't see a resource, I saw life. And my instinct was that the microscope offered me a privileged insight into this otherwise unseen organism. Here I could see them moving and living in all their abundance and reflect upon their origins, not their use. So I wanted to create an installation that gave a more embodied sense of this microbial other, something that enables us to see and also hear these organisms, playing on what evolutionary theorist Lynn Margulis called science's general impediment to perception. So I worked with Dr. Paul Vickers, a computer scientist from Northumbria University, and Dr. Richard Thompson, a material chemist from Durham University, to listen to these microorganisms, which we did by means of an atomic force microscope. By placing the liquid medium containing the Dunaliella in the fluid cell attachment of the atomic force microscope, we were able to record the data picked up by the microscope's laser probe as the Dunaliella propelled themselves through the liquid. So it was then possible to audify this data by translating the fluctuating values into frequencies audible to the human ear. So for Lumiere, the audience could see and hear Dunaliella. the view that we presented is not any less true. It's simply another perspective that enables us to reflect differently on the life of the microbial other. Following Lumiere, I wanted to expand this perspective of the microbial other by presenting them as they inhabit space alongside us. As part of this event called Domestic 2 in Manchester, sound artist Mark Reed and I were part of a group of artists given access to individual flats within a tower block in Salford that was about to undergo renovation. We visited the site in advance and took samples from within the space and cultured them, even grew some sourdough from the yeast that we took from the air, and then reintroduced them, making live recordings and projections within the space. The title, Natura Naturans, means nature naturing, or nature doing what nature does, a phrase termed by Spinoza to refer to the essence of something. And so the aim here was to presence the microbiological for the viewer, again, altering our perspective. But whilst this work enabled me to gain a deeper experience of the microorganism, I had not yet found a way to experience our impact on it. Microorganisms exist all around us and even within us, and I wanted a means to explore how our interactions with them at the technological level might impact our broader symbiotic relationship. So, last year I began working here at the Institute of Genetic Medicine at Newcastle University to pursue my interest in humanity's manipulation of living matter. I'm sure many of you in the room are aware of the field of bioart, and even outside of this relatively new terminology, a number of artists have worked with DNA or with life in some form, living organisms. So again, coming back to Heidegger, I'm questioning concerning this technology, this biotechnology. I'm interested in what Heidegger referred to as a hermeneutic circularity, and that to me is the problem of technology as distinct from nature. Technology is, in Heidegger's words, a revealing, and modern technology is a challenging, and I think that now with biotechnology we enter into a third possibility, and that is a folding back upon. Biotechnology has the potential to disclose aspects of our own nature as living beings in a manner that might almost be termed cannibalistic. So in my research, I used the techniques of biotechnology to explore what might happen if synthetic information is stored within a living organism that is then granted the potential to evolve. What will the essence of life make of our actions? Can there be such a thing as synthetic evolution? So, I've created DNA that does not, to our knowledge, exist in nature. And I've inserted this within a living organism, the synthetic biology vessel of choice, E. coli, to determine whether it is possible to store information in this manner without impacting the evolutionary trajectory of the organism. I'm using life as artistic tool and evolution as my methodology. As an artist, I wanted to find a way to make this supposedly reducible component of life speak to me. I wanted to listen to evolution. And I chose to do so using a medium that I felt offered the complexity of evolution inherently, yet was familiar enough for change to be recognisable. I was inspired by Alvin Lucier's piece, I Am Sitting in a Room, in which he recorded his own voice within a space over and over again. And so, in the tradition of other artists who've worked with transgenic material, artists such as Eduardo Katz and Joe Davis, I've created a cipher that in my case would enable me to hear mutation of the spoken word. So the cipher converts standard English language phonemes to, plus some additional dialect phonemes that I've added in there, into codons, the base components of the genetic code. So the codons build into amino acids, which build into proteins, which build into the structures that form life itself, and the phonemes form into words, which form into sentences and the complex structures that are language. And I like this parallel as a means to view evolution as some form of conversation over deep time. So using this cipher, with the help of Dr. Anna Topf and Dr. Steve Laval at the Institute, I was able to encode a phrase within the body of an E. coli bacteria. I spent a long time thinking about the phrase that I wanted to encode, and after a while realised that it had to be a question that I asked my microbial colleagues. I am, after all, looking for an answer. Perhaps not in my lifetime, but I'm attempting to reveal within them a sense of agency of their accumulated knowledge over time, passed down through generations. And so the sentence is a shortened form of this text, and it's a question: what will happen if I store this thought safe within you? So the sentence has been recorded as a spoken phrase, and the individual phonemes have been translated into codons that were created as synthetic DNA stored within a plasmid, which I then transformed into E. coli bacteria in the lab. What's significant about this for me is that in manipulating life, I become responsible for its evolutionary trajectory. I've created something that did not exist without me, and therefore I feel this sense of responsibility towards it. And on this website here, a little plug, there's a short documentary-style film which explores my views on this, my way of working in this way a little bit further. So if anyone's interested, that's about 4 minutes long. You can watch that in your own time. So far, I've been nurturing the E. coli for months now, and the phrase has remained very stable within them. And with the assistance of Amino Labs, who developed the world's first portable biolab, I'm creating a nurture device in which I can look after these living organisms of my creation indefinitely, so that I'll be able to sequence them and periodically check their DNA for mutation. In addition to the in vivo experiment that I've been explaining, I've also created an in silico version of the experiment using bioinformatic predictions of E. coli evolution. And with the assistance of Edinburgh University's Department of Speech Synthesis, we're building this into an audio installation. I've tested an earlier version of this, and I have a brief demonstration of some of the mutations for you here.
What will happen if I store this thought safe within you? What will happen if I store this thought safe within you? What will happen if thought safe within you?
It just gives you an example of the way it can change. So the inclusion of a synthetic voice was actually important to me in referencing this hermeneutic circularity. Will synthetic DNA lead to synthetic evolution? What might it mean for the future of life? We tested a mock-up of this in silico version of the experiment at an installation at Baltic 39 in Newcastle for the late shows back in April. I worked the synthetic DNA construct that we had into a physical clay vessel, and its predicted evolution within E. coli was sonified through an 8-channel audio into the surrounding space, creating a sort of increasing cacophony of disembodied human-like voices. And I likened this spilling out of genetic truth to the pathos of the Pandora myth, the original curious animal. And I'm now working on a larger installation which will contain this piece, a multi-channel sound installation, along with a sculptural work housing my living E. coli, a film of my ongoing work to sequence them, and some software that will enable audio readouts of any mutations as they occur, which I'm tentatively calling the genophone. In pathos, I'm allowing an organism to live and thrive with synthetic information held within its DNA, and I'll monitor it throughout my creation's many generations, noting any changes. I'm not interested in editioning them as artworks, nor is it my desire to make a living artwork that I must then kill, although they will inevitably die. My interest lies in the creation of a new species via synthetic selection. Part artwork, part life, and as such this species has the potential to evolve as an art-bio hybrid species beyond my own life expectancy. I do so to question our ability to impact evolution, to demonstrate that with synthetic biology we also see the advent of synthetic evolution. Arguably the artwork has a right to life and its own evolutionary trajectory, so therefore I question whether it should have been created in the first place. Related to this work, my colleagues at the Institute and I are developing ideas for a new piece. The Laity Without Volition, which is its working title, is the physical manifestation of human will in a biotechnological era. Using the same synthetic biology techniques I've described already, we plan to engineer a string of synthetic DNA that describes a thought from my mind to be placed within my own biopsied skin cells. This thought is volition, a will to act, held external to my mind, yet internal to my body, or at least my cells, as synthetic other. The thought will be transcribed within my cells through a viral host, which infects my cells, imposing my will upon them. It becomes at once part of me and alien to me. I'll then nurture this externalization of my body to determine whether my cells accept, reject, or alter this synthetic will. The ultimate aim is to reintroduce the cells containing my will to my physical body. So really, this work questions our intentions for the future of synthetic biology. Will we stop at genetically modified food, mosquitoes, or the production of synthetically healthy cells to help cure disease, when we can pose our will in more creative ways? What might it mean to store information within our DNA? What happens when we commoditize our cells as repositories for our use? Can we really create living machines that will better humanity? Or are these claims by their very nature of velleity, a wish that we have no strong desire to follow through on? Buffered by substantial investment into such wishful thinking, the field of synthetic biology grows apace, with no body fully accepting responsibility for the unknowable long-term outcomes. And it is my will to circumvent this velleity by using my body as site of responsibility for the imposition of will. And that's the end of my talk. Thank you very much.
Wow. Okay. Questions? I'm going here first because you missed out earlier.
You were talking about introducing your thought through a virus into the cell.
Yes.
I was wondering what your thoughts are on using some kind of CRISPR-esque technology in introducing it into the genetic code itself rather than just as a cohabiter of the cell.
It will be part of the genetic code. So using the same sort of process that we've already done for the E. coli work, we're going to create a piece of DNA that will be part of the virus, and then the virus will impart it directly into the genome.
Thanks. It's always nice to hear somebody else is working on the extinction of mankind by putting genetic material into bacteria and viri. Do you have a number of probability that artistic experiments like this will actually release something that will kill us all?
Well, I suppose the reason I'm making the work is that I'm much more concerned about the possibility that something like that can happen, whether it's by an artist or anybody else, as the world of making with life becomes so much more open source. And so really I'm exploring it from that perspective.
So if it's going to happen, you want to be the one who's...
Well, everything that I'm doing is very much done within a laboratory environment and contained and controlled. And I'm actually, up until now, only working with what's called Class 1 genetically modified organisms, which in the US are not even considered hazardous. Here they have much stricter controls over that kind of thing, so I'm still very much within the bounds of the laboratory, although I'm looking at ways of at least taking these genetically modified organisms into another context, albeit with the relevant safety measures in place. But there are a few other artists who have done it in the UK. Howard Boland, for one, has held an exhibition of that nature.
Andy.
It's interesting when you saying synthetic evolution. So, how are you thinking that these injections might alter evolution of your organisms, and what are your thoughts on
Well,
evolution?
that's the part of the joy of it is that I have no idea what will happen. The first work that we did with the plasmid was with the plasmid introduced into the E. coli was very, very safe, and the likelihood of any change is incredibly minimal. But that's not to say that a change won't occur. And so that's why we ran the predictive modelling to see how change might occur, and that's why I want to keep the experiment long running to see if any change will occur. But the types of change and whether the plasmid is somehow either broken up completely or whether it is somehow becomes incorporated into the E. coli more fundamentally, all of those things are, I suppose, possibilities, but we just don't know. So it's part of the work, really.
Any others? Yes. When you were going to ask a question.
That's a quick one. I'm just following up on that, really. I'm not quite sure. I mean, isn't this just ways of introducing mutations into the organism? I mean, I'm not quite sure what the evolutionary bit is. I mean, I'm not quite sure what the evolutionary stages are. They meant to, in some way, create new variants, new species in some way. I mean, I can see that you're introducing synthetically alterations.
Yeah.
But that's just, I mean, isn't that just mutations then? I mean, in what way? There's mutations everywhere, and a lot of them completely scupper the organism, and there's no evolution. It just falls out.
Well, I suppose what I'm interested in is the notion of the initial insert of information being entirely synthetic, i.e. it's an insert that we have introduced, it's an insert that a human has introduced into the organism. And so, yes, I'm not looking to force mutation. I don't want to force change. I want to know how a living organism responds to having a piece of synthetic information inserted within it. So it is, I'm specifically looking at evolution as opposed to some more forced mutation, let's say.
Could I just ask another thing about the translation? I can see how there might be some way of devising a code to translate genetic information in some form into linguistic forms, items. I'm not quite sure about the other way around. I mean, I can see you can sort of do it. You could generate some bothersome mutation that you could introduce, but it doesn't really translate into anything other than just some kind of intrusive mutation, because it doesn't really express anything, does it, at the end?
No, it's not. We're not actually aiming for. When we created the initial sequence of synthetic DNA, which basically the way we went about that was that the mapping. I don't know how much time I have to go into detail, but essentially the mapping was the 64 different codons in the genetic code. I took the standard, I think there's around 44 phonemes in the English language, and I added different dialects as well, so that we had a direct mapping. And then we worked by sentence. We basically found a way to make that mapping such that I had a sentence that would not express a protein, because we didn't want to create anything that would potentially cause anything difficult to happen. But that is something that we could think about doing in future work, and I know that there are other artists who have in fact deliberately done that. Christian Bock, I don't know if anybody's aware of Christian, who's a poet who has specifically coded a poem into DNA such that it deliberately expresses a particular protein. Whereas what I want to do with this work is not force any form of expression, but to see what the natural living entity does with the information that's imparted within it. So it's quite a long-running piece of work, essentially.
Okay, thank you very much, Louise.